Welcome. This is the invasive species track. If you're not supposed to be in the invasive species track, you probably should be down at one of the other buildings down the road, so you might want to head that way now. My name is Carrie Brown Lima, and I'm the director of the New York Invasive Species Research Institute, which is based here in the Department of Natural Resources here at Cornell. This track has been going on for over a decade, maybe even 12 years, and is grown so much. I'm so excited to see this room so full. It's actually doubled in size at least. And this shows the great advancement of invasive species in New York State, and also just the network of CC educators that are interested. So we have over 120 people registered, which I'm so impressed with. And we will have people moving in and out. Not everybody's going to be in this session. So if you have any questions about where you should be going from here, there's a detailed agenda inside your folder with your sessions on it. But we also have a separate agenda that's just for this track that will tell you more about the speakers and about the sessions. So if you haven't grabbed one at the table out front, do so, or Audrey, you can pass them around later for people who need it. And just a little bit on logistics, there's also some other materials about the New York Invasive Species Research Institute, and also about some of the different projects going on. We have spotted lanternfly information out there, a pen drive on biocontrol, so check it out so you don't miss out on any good information. As most of you have discovered, we have food and snacks in the back, and we'll have those throughout the session, a morning session, a morning break, and an afternoon break. And then we'll be breaking for at least an hour and a half every day for lunch. Bathrooms are out this door in a big U, and they're actually over here. If you really want to get there fast, you can cut through the kitchen or the closet, but I think you're supposed to go around. And I really just want you guys to feel relaxed. If you need to get up and get a coffee or stand up in the back, because I know it's a long time to sit, please feel free to do so. We have a really packed agenda and a great lineup of speakers, and we really want this to be an opportunity for you to learn from our speakers who are bringing great experience and information, new techniques, new ideas, and we also want you guys to share, because one of the great things about the in-service and what it's supposed to be is an opportunity for us to share what we know and learn from each other. And so please don't miss out on that opportunity to take advantage of the breaks. Please feel free to ask questions. We've tried to build in as much discussion time as possible in each session, and please use that. It's not just to ask questions, it's also you can share information about your projects. And I just wanted to make a few thank yous here to Melanie Moss, Audrey Bowe, and Sarah Deegan-Muzio, who have helped a lot in the coordination and really made this happen. And before I introduce our guests, I just wanted to say this is a 2-hour session, and from here we're going to go to lunch, which will be on your own. So there's a dairy bar here. There's other places on campus that many of you are familiar with. If you have any questions about where you should go or where you can go, ask me or Audrey or anyone that's at the table. But before you leave for break, we've never done this where we've actually gotten a picture of our group, and we have such a great turnout of diverse partners from across the state that we'd like to just gather up for 1 minute and take a quick photo. So with that, I will pass the mic to someone who has a lot more interesting things to say than talk about the bathrooms and the snacks. Our first speaker today, I'm really excited that he could make his way here from Bozeman, Montana, is Dr. Pete Coppolillo. All right, great. He's the executive director of Working Dogs for Conservation. This is a topic that I have really wanted to explore more here in New York. We're always talking about high-tech early detection techniques. This is a low-tech one that's completely underutilized, and I'm really excited for us to learn more about it today. He's also come together with Amy Hurt, who's the operations director for Working Dogs for Conservation, and then afterwards we'll hear a little bit from here in New York, a new project that's actually training a dog for conservation. So Linda Rollader and Josh Beese will be talking a little bit about their experience. But I'll start off by passing the mic to Pete. Thanks so much.
Okay. I don't want to blast you guys. Is that too loud or is that okay? Okay. Thank you. Thanks for having us, Carrie and everybody who set it all up and everything. And maybe Amy and Josh and Linda, you guys just raise your hands too. As will become obvious, I'm kind of the talking head for our organization. Mostly what I do is chase the money and direct traffic within the organization and set up new kinds of partnerships and all kinds of new things that dogs do. So coming to venues like this is really, it's a huge opportunity for us because we do, as you'll see, this talk today is pretty much only about invasive species and a little bit about disease and biosecurity, but we do lots of different stuff, law enforcement and endangered species and all sorts of stuff all over the world. So it's hard to really track and know what is really needed on the ground in the field. And you guys are out there every day, or you're probably like me, got trapped behind the computer and supervise people who are out there every day. But this is as much for us to collect some information and know what's going on. So in one of the first pieces of literature I picked up, I immediately handed it to Amy and said, hey, we ought to be working on that. So I'm just as interested to hear from you guys about all of that stuff too. So as I plod through this, there should be plenty of time. We've got lots of time. So if I'm not being clear, or if you want a little more information on something, you feel free to interrupt and stop me. And if we're going to get to it, I'll tell you that. And if not, I may defer to Amy or others and just ask. So we'll just sort of dive in. And I guess tomorrow I'm going to do a talk for campus, a more general one. But I thought I'd just give you guys just a brief snapshot about where this field came from. And I'm a conservationist. I'm trained as a landscape ecologist, and I came to it through the conservation side because I kept hiring these guys. And I'm sure this is a familiar story to you guys. As conservationists, we're always focused on rare stuff that's declining, stuff that probably even before it started to decline was never really all that abundant. And so, we work on a lot of species like these that are you can spend a whole year in the field, or even some people multiple years or a career without ever actually seeing them, right? And so, as biologists, we tend to up the ante and up our game, which tends to be more invasive and more expensive. This is just one example from my past. A pronghorn antelope out on our side of the continent is actually a pretty abundant thing, right? We caught 72 of these animals. We were looking at a migration in and out of Teton Park. Caught 72 of these guys to fit them with radio collars in a day and a half. It cost over $220,000. We spent in 36 hours. And, it's expensive. It also can be dangerous. I choose this example because pronghorn operations, we were very good. It was a fabulous team. The helicopter and the pilot and the net gunner were amazing. We didn't lose an animal. We didn't injure an animal. But historically, pronghorn operations can lose mortality up to 15% of individuals are handled. So it can be in addition to being costly, it can be dangerous for the wildlife. It can also be dangerous for the people. This is the same helicopter that we were in. That was 3 weeks later. They were over in Idaho on a wolf project, and it was a mechanical failure. They were very lucky in that everybody survived. You can see they didn't walk away, but everybody did survive. So it can be really dangerous for people as well. And just so you don't think that I'm being hyperbolic, this was a few years previous. That plane was being flown actually by one of our co-founders. That same year, they were radio collared African wild dogs. So they were going up to get a fix in the air. That same year, 3 other airplanes hit giraffe in Africa. So it's a strange phenomenon, but flying around is dangerous. It's expensive. So about 20 years ago, Amy and the 3 other women who co-founded the organization all kind of came together and said, there's got to be a better way to do this. We need something that's more reliable, less expensive, safer for wildlife, safer for people. And this is where they ended up with it's a dog's nose. And it's not an exaggeration to say that a dog's nose really is the best chemical sensor the world has ever known. And the reason for that is because it's so flexible. They can do an extraordinary number of things. They're really good at discrimination, telling two things apart, as well as detection. So I'm going to give you some of the examples about this, but the best part about them is that they're so flexible, and they're so interested in working with us. And it's kind of a funny story. It's a little bit embarrassing to say that it wasn't until the 90s that we started doing this kind of work with dogs, because dogs have a very long history. The closer we look at it, the longer we realize that it actually is. You know, estimates now are, you know, people say 40 to 50,000 years we've been living with these carnivores. And of course, we all know that carnivores run around the landscape leaving little messages for other carnivores. And it took us until the 90s to say, hey, why don't we use these ones to help us figure this stuff out and find them. So it was in the 90s when some of the first conservation detection dog programs happened. It was with bears, grizzly bears. And part of what catalyzed the whole field was developments in the lab. So like Carrie said, this is old, low tech, but one of the things that really facilitates and drives our field forward is the high tech, the stuff that happens in the lab. So back then, getting DNA, just to get DNA to say it's a grizzly bear, not a black bear, that was really expensive. You guys all know this story. Now it's cheap. And not only can we say it's a grizzly bear versus a black bear, you know, we can say it's Fred from Idaho, and you know, he's the son of, you know, M42 and, you know, M60 whatever, and we can also look at stress hormones, reproductive hormones. We can look at diet. You know, the quip that I always make is the value of turds just keeps going up and up, right? And dogs are really good at finding them. So as the lab costs go down, the more we can do it. So there's lots of stuff to do that way. So the early part of the field was looking at threatened and endangered species. That's a grizzly bear in Yellowstone. And you know, grizzlies leave very big scat piles. They're very easy to find. So Amy and the other founders really started pushing the envelope and seeing what else we can use dogs for. This is a San Joaquin kit fox. They've been on the endangered species list about as long as there's been a list. They have the misfortune of you know, historically being not that abundant, not very widespread. They live in the southern San Joaquin Valley, where now they are facing habitat loss. Primarily, you know, back in the day there was agriculture and some recreational development, and now they're looking at a lot of renewable energy development. And so under Section 7 of the Endangered Species Act, you have to monitor the population. You guys all know this story. And these guys are incredibly shy. They're underground in burrows most of the day. They don't stare at a spotlight. They're in a scrubby environment anyway, so they're hard to see. They're little. They won't come to track plates. They don't rub and do hair snares, things like that. So finding their scats and analyzing them that way is about the only way to really get reliable estimates. And it certainly is the best way. So we work now in the largest solar, or what will be the largest solar farm in the world, counting kit fox. And it allows that project to go forward. And just as an aside, it's a really cool story, and they're doing quite well. So it's a nice one to take, you know, climate-friendly energy and make it wildlife-friendly as well. As we've expanded, as it's gone on, we've also expanded the range of species that we work on away, you know, beyond just carnivores. That is a camera trap photo in the upper left there. That's the Cross River gorilla. It's the rarest great ape in the world. It's like a mountain gorilla, but they live just only on the border between Cameroon and Nigeria, highland. It's an incredibly remote area. That's why they're still there. So remote, in fact, that you can't just walk in there. There are cliffs, and had to carry Lily in there up and down to get there. From a technical point of view, gorillas are easy. We joke they're cows with opposable thumbs, right? They leave plenty of scat behind, but even the villagers who live adjacent to the forest where these gorillas live never see these guys. So what the dogs made it possible to do was not only to census those areas, to identify the forest fragments that are occupied, to know how many individuals in there, but our CDC and North Carolina were involved in this, they surveyed the scats for both human and gorilla diseases as well. And this was in between some of the Ebola outbreaks that may partially explain some of the distribution of those gorillas, but also have big human health implications. And then it's continued to expand to all sorts of other species. This is actually a bit of an odd one, a strange one, but our National Park Service came to Amy and asked, hey, we've got all of these crevice nesting bats, and we don't know which crevices they're in and which crevices are not, and so that really hampers our ability to say what should and shouldn't be allowed here in the park. So can you help us find that? And we said, I don't know. So Amy and Lily there too. You can see Lily's done a diversity of different things. You'll hear more about her later because she's really kind of anchors a lot of the invasive species work. So this is in Iowa, checking crevices for smaller roosts and for bats. So you guys are scientists, so I'll give you a little bit of data here. This is just a summary. A few years ago, we went back and said, what are dogs good at? When should you be using them and when shouldn't you? And the bottom line is that they're good at stuff that's either cryptic, hard to find, or hard to discriminate, or at very low densities. So the 36 more likely than hair snares to detect a single individual. So if you've got really low densities, you can camera trap for a really long time, and it'll take you a long time to find them. Whereas a dog who's searching that area systematically will find it a lot more quickly. Turtles are a nice contrast to that. There are plenty of turtles around. They're just really good at hiding, right? So dogs can be really good at finding those things. So as the field progressed, this was the kind of stuff that we started doing. A lot of from carnivores to a broader suite of species to some live animal work. But now I'll tell you, sort of give you the thread that ran through it to get to the invasive species work. And this is one of the beginning points for that story, which is it's a Fender's blue butterfly. And it lives in Oregon, and it lives on a particular species of lupin. And I can say with a great deal of authority that I am probably the worst botanist in the room. I can't tell Kincaid's lupin from another species of lupin. Even people who are good botanists have a hard time doing that. So the question was, can dogs do it? And the reason for doing that is that's the host plant for Kincaid's or Fender's blue butterfly. And so we asked, well, I don't know, let's see. So through that project, they ran 5 dogs, 378 trials, and they only missed 6 times. So the upshot of it is, yeah, they're really good at that. And just because we can't see a difference doesn't mean they can't smell a difference. And so a lot of times people always say, well, how do they smell that? And I say, well, we dedicate a small fraction of our brain to vision. Dogs dedicate a much larger portion of their brain to olfaction than we do. So if we can see differences, almost certainly dogs can smell differences. And so the discrimination side of dogs, we talk a lot about teaspoons of sugar in Olympic swimming pools and all of those very low density detection tasks that they do. But one of the really most important and best things that they also can do is to discriminate things very, very accurately and also very quickly. So that led to some work on spotted knapweed. And this is Savo. He was one of the early knapweed dogs. And flagging and having known distributions of plants allows us to then run dogs through there and determine detection distances. That can help us do things like space transects. Depending on what the objectives are, do the transects need to be close? You want to find every individual. Here's how they should be. Occupancy or not, or all of those sorts of things. And the bottom line that came from that work was that the dogs were good, even better than people, at finding spotted knapweed. So again, not a big surprise, but very useful to help start put numbers to it and to understand if you're going to sample, how would you go about doing that. So that's kind of all the background. And now I'll tell you, just share a few more stories with you on where things are these days and the kind of different contexts that we do the invasive invasives work in. And those are just three. I used to always say dogs are a first plant, last plant. So detect the first one that's there, or hopefully the first one that would have started an infestation, and to help let you know that you got the last one when they're done. And what I'll share with you in the middle is now there's beyond the first plant and prevention and the last plant, eradication, we're doing a lot more in the middle on either management or mapping. So at kind of all these different stages. So I'll tell you a few little stories about those in the middle. But this first one is prevention, and it is the most promising and in some ways the most difficult part of this story. So I'll go through sort of each of these examples. This one you guys unfortunately are very familiar with. It's zebra and quagga mussels. In our part of the world, they may have just arrived or may not have just arrived, but you guys know the story. It's a bad deal once they arrive economically, and they move very quickly, and the primary vehicle for them is boats, of course. So we were at a conference in Montana, gosh, I don't know, five or 6 years ago, and talking about zebra and quagga mussels and veligers, the invisible larval stage. And what does it matter if we're inspecting boats or draining tanks if a boat's covered in veligers, if it's just going to infect a waterway? And so a fellow who was Ted Turner's aquatics person said, well, why don't you train a dog on those? And we said, I don't know if you can do that. So we decided to see if it was possible, A, to do mussels in the first place, once they're dry and closed up and all of that, and then B, to work on veligers. So we don't have mussels in our part of the world. So Amy, and that's Wicket there, had to pack up and head to California, where they do have an active infestation and where it's okay to pull them out and mess around with them and do all of that. And so started with a variety of small boats to detect, started training in a sort of lab situation, which I don't show here, and train the dogs on, this is the scent you're looking for, obviously, and then check it on actual boats. And no big surprise there. The dogs were quite good at it. Interestingly, the feedback that we get from the officers at the check stations is, yeah, it's great that they're so good at detecting them. What's really great is how fast they work. So, we have a mandatory check station law in Montana, which people sometimes obey and sometimes don't. But when you get 2 or 3, like a big pontoon boats and things like that lined up, it's going to take that inspector an hour or more to really thoroughly go through that boat. Well, a dog can do that same boat in about 3.5 minutes. And so when people start getting angry and stroppy and not wanting to sit in line, they want to go get their boat in the water, and the inspector starts saying, just go ahead, Because even in an active infestation in California, the rates of detection on boats are about 1 in 1,000 boats anyway. So for them they'll just start waving them through. But when there's a dog there, they're moving much more quickly, and people are interested to see what's going on. What's the dog doing? Now, in certain parts, I don't think I put a picture of it in here. There's a signboard that goes up that says, here's what the dogs are looking for. It's zebra and quagga mussels. Here's what they're not looking for. Guns and ammunition and your drugs, just fine. Leave your stash in the boat. It's no big deal. So nobody blows through the check station worried about that. So the dogs did a great job looking for live mussels and planted mussels on the boats. And then in a more difficult, and Amy can give you the details about this, and in a contrived sort of setting, they also demonstrated that, yes, they can detect veligers in water in very low concentrations. What was the lowest one, Amy? 12 individuals in
2 in 12 liters of water.
2 individuals, 2 veligers in 12 liters of water, and they could reliably detect that. So they're quite good at it. Even when it's uncommon. So this is a pretty active area for us, unfortunately. and I'm going to return to it in a little bit, and how there's a whole institutional side of it that's an interesting story that is worth sort of thinking about as well. Okay, the next sentence is a similar sort of project in that some of you may have heard about it. They got a lot of press. South Georgia and the South Sandwich Islands, they spent 10 million pounds getting rid of rats on those islands. And the reason they did that is because those are some of the biggest, they're amazing seabird colonies there. And of course, they've evolved to nest on the ground. And so things like weasels and mink and mongoose and rats, if they get in there, they can just wreak havoc because nothing's adapted to dealing with them. So, not surprisingly, rats were introduced to the Falkland Islands and to South Georgia and South Sandwich via boats. And the British government and a couple of charities came up with the money to get rid of them. It was an amazing effort. And I'll show you a picture there were dogs involved in that effort. Different dogs, not ours, but I'll tell you a little about them. And now they were successful. And so they said, never again. Interestingly, with climate change, some of the glaciers on South Georgia and South Sandwich Islands have retreated enough. They've created, in the Scree and Talus slopes that left behind, they've left refugia for rats. And they said, we will never have this opportunity again. If rats get back on those islands, we won't be able to eradicate them. So they said, we have to be 100% sure. So down here on the bottom are Colonel, who's a Beagle mix, and Samurai, who's a Shiba Inu. Not our normal kinds of dogs. We have lots of Labs and Belgian Malinois and big, hardy dogs. And these two little yappers went down there to go after the rats. And the reason we use the little ones is because we have to lower them down. They have to be carried down galleys and into the hold of ships and get into very tight, small spaces and to do that kind of work. So last year we piloted this program for the government of the Falkland Islands and South Sandwich Islands. And this year we're taking Samurai and a new dog named Brody. We'll go down and hopefully start the permanent program for them. And every boat that goes to those islands, there are only three reasons to go there. One is tourism, one is research, and the other is fishing. And the tourists and the researchers, they want to do it right. They're happy to wash their shoes and to do all those things. It's tough to get the fishermen to care about rats, and fishing boats are pretty nasty places, a pretty good place to be a rat. So that's where a lot of the action is and will be. So they'll go back down and start that program. And a lot of the program is not just the technical detection, although it is an interesting problem because, if I can be a bit crude, everything on a ship has been pissed on from a rat at some point. So you can't train on urine. You have to train on live rats and feces. And so it creates a technically a little bit difficult challenge for the trainers. They work that out and have figured that out now. But the challenge would be to go back and to fit all of this into the workflow, right? So the boats are not always at port for a very long time. So they have to get on, do the inspection, and get off, and then do the notifications. Because you don't want the captain of the ship kind of coming along and curating where the dog goes and what he looks at, right? Or saying, oh, no, that wasn't an alert. Or, yeah, there was a rat in there, but it's gone now, or something like that. So the workflow is just as important as the technical detection dog side of things for that one. Okay, this is another beast that I think you guys may be familiar with, emerald ash borer. Amy was part of a program now 6-7 years ago with Minnesota. And again, this is another one that's a little bit technically difficult because you can have the adult, but you can also have larvae or eggs. And there's a bigger sort of wider problem is, what do you inspect? You can't inspect every piece of wood every time. So for that project, we ended up training different dogs to discriminate ash wood from other woods, even when it's in mulch. And so that was a bit of a sort of breakthrough, because then you can say, these are the things that need to be inspected, these are the things that don't. And that's a very similar process to looking at illegal hardwoods in Africa. A lot of times they will, like I said, we've been doing a lot of law enforcement. They'll just ship, once they're sawing logs, they'll say, well, nobody can tell what that is. And then they'll just, openly sort of ship it, but a dog can discriminate, can say this is this, this species versus that. So again, the bottom line is they were good at it. They were good at discriminating the woods and to detecting all three life stages of ash borers. We got a lot of requests after that project ended. Unfortunately, the next phase in the permanent implementation of this was in the agriculture bill, and that was the year that, over political infighting, the ag bill died and never got funded. So unfortunately, that project just sat there. Since then, we've had a number, as ash borer has moved across the West now, we've had a lot of people come and ask, can they do it in trees, actually in urban forests? And it's an interesting question. It presents some really difficult challenges for a scenting dog, and what do you do when they do detect it? My intuition is, yeah, they'll be able to do it, but the question is, what will we be able to do with the information? But we haven't actually done it yet. So that's another one that's sort of dangling out there. Okay, I mentioned this sort of middle phase, which is the management or mapping phase in the middle. And that seems to be increasing for us as well. So up on top, feral swine, Argentine ants, and the bottom is brucellosis. I'll mention all of those, but just recently, well, the first picture here, this is actually Lily again and Amy. This was just last week, looking for settled mussels in a reservoir in Montana called Canyon Ferry. We had 2 years ago, a DNA sample, water samples from 2 water bodies in Montana came up positive for mussels. No one has yet confirmed mussels in either of those areas. So this time of the year, when the reservoirs are drawn down as low as they're going to go, Amy and colleagues from Alberta, with dogs that Amy trained, have pioneered really a method for shoreline searches to say, if it's here, this is probably where it's going to be. Let's see if we can find it. Interestingly, we have not found it yet. So they're sort of waiting out the 5-year window. Maybe it didn't take. Maybe it was a contaminated sample. Maybe there was just a little DNA fragments from somebody's boat. Wasn't there? We don't know. But the shoreline searching method is brand new. And as you guys know, these are really complex environments. There are a lot of places to hide a mussel around a reservoir. Canyon Ferry is I don't know, 11, 12 miles long, and like many reservoirs, has a very complex survey. So it's sampling. They're just picking spots where to look. And of course, a lot of it, one would guess, will be associated with human infrastructure where boats are coming and going at the access points. So that's an interesting one, sort of to be continued. We'll see if it turns up. We will continue monitoring. Unfortunately, our legislature is keen to declare victory and stop funding it. So maybe is keen to stop funding it and then ensure that we'll be doing that work again later on. We'll see. Okay, a second one is the Channel Islands. This is Deb Willett. She's also one of the founders of the organization, and Kai Zimmerman. He was working as a boat inspector for Fish, Wildlife, and Parks, got excited about the dog program. That's another yellow lab. It's not Lily, it's Tobias. He was rescued from Helena. And they are on the Channel Islands in California working on Argentine ant eradication. So the Navy and the Nature Conservancy primarily have spent in the millions also to get rid of lots of stuff on the Channel Island, goats and pigs and Argentine ants. Many of you may know Argentine ants if you've lived in California, particularly in the winter when the weather turns like this and they all come inside. They're the tiny little black ones, and they're in everything. And it's an interesting story for geeks like us because it was, in Argentina, the colonies recognize the difference between each other and they fight and kind of keep each other at bay. When they came to California, because of the founder effect, the whole population in California thinks of itself as one colony, and so they don't hold each other back. The colonies get huge. It's a giant super colony. They're everywhere. So mainland, I haven't heard anybody talking about eradication on the mainland, but on the islands, it is a possibility. Tobias was brought in to do 2 things. One was confirm that on one island, confirmed that the eradication was complete and was finished. And it turns out it wasn't. He found 3 colonies that they had missed. And on another island, he was brought in to map the outer edges of the infestation so they can start that eradication project. So it was kind of a cool one. It was the end, and in a separate spot, the intermediate sort of stage, so that they can target all of their work to do the eradication more completely. This is a little stretching in terms of invasive species, put it more in the biosecurity sort of category. This is brucellosis. This is a big deal in our part of the world, in Greater Yellowstone. Brucellosis is a Eurasian disease that affects cattle. You guys probably know this story. In the 20s, when we had only a couple dozen wild bison left in North America, they were in the Lamar Valley in Yellowstone. And the calves were bottle fed with cow's milk before brucellosis was eradicated from North America. Well, in the intervening period, it was eradicated everywhere in North America except in our bison. Bison have now transmitted it to elk. Elk are the agent between the wildlife reservoir, which is, as far as we know, just elk and bison. The only transmissions that have ever been recorded have been from elk to cattle. And you guys probably know this, bison wander around in a big group. Their prevalence is very high. They don't interact with cattle very much. It's not hard to know where they are or what they're doing because they're such herd animals. Elk, on the other hand, very migratory. They're like ants. They're all over the landscape. They fission at different parts of the year. They come together. And the state of Wyoming has this kind of crazy practice of feeding elk. And so they bring them together for these tremendous disease transmission events. So every winter. And so that's what the dots are. And as you can see, brucellosis is spreading in our landscape. At least we think it is, because every time we start looking farther and farther out, they keep finding it. So we spend between $12 and $16 million a year doing that, looking for brucellosis and mapping it. So we got to talking about, well, can dogs help with this? So every year they capture a whole bunch of elk on the feed grounds, and then they take blood and fecal samples and all sorts of stuff. They send them away and do the get the titer numbers for each one of those animals. So we said, hey, can we have some of those pellets, the elk pellets, the fecal material, and then when you get the numbers back, can you tell us who's positive and who's not? And then we'll see if the dogs can discriminate an infected elk, the scat of an infected elk, from the scat of an uninfected elk. It's a funny process because they're high and low titers. It's not a 0 and 1. So we just have to arbitrarily cut them off. We say 0.8 or above or 0.2 or below. Those are we call them positive and negative. And it turns out with over 80% sensitivity, dogs can discriminate between them. So the next stage of this work, that's as far as we've gone, was a lab study. The next stage of this work will be to actually go out into the field and to map it. There are a lot of things that need to happen between now and then. The University of Montana is working on isolating, getting DNA from, or a lab technique to verify brucella in the scats. Because if a dog alerts, we pick up the scats, we need to verify it some way. So I'm going to return to that idea of verification in the field because it's really important. But that's one of the things that's hampering this. But we'll see where we can go in the future if dogs might be able to help map those boundaries and then use that for extension to say to some ranchers, hey, you need to keep your cattle out of the field until June because that's when abortive fetuses, which are from elk, are the transmission agent to cattle, and things like that. So this one's kind of work in progress. I have a lot of hope for it. It's also a model. You guys are probably talking in this part of the world about chronic wasting disease now too. It's in the margins. It's at the edges of our little world in Greater Yellowstone. It'll be in there relatively soon. Okay, so the last one is our eradication efforts, and this is I think it's some of the coolest and most hopeful sort of stories that are there. This is one of the dogs who was involved in the eradication on South Georgia. He came from New Zealand, where it's a fascinating history. New Zealand and the United States. New Zealand's been using dogs for for conservation for a long, long time in a very different way. And a lot of it is invasive species work for reasons that you guys understand far, and know the history of and far better than I do. So those dogs came. The big misconception about dogs and eradication is that the dogs are going to kill the rats. The dogs might kill a rat or two, but dogs are going to get hurt. Rats are probably not going to, and you're never going to get every last one. And you're going to annoy a lot of people, right? So it's not, people say, well, if we have a feral cat problem, can you just bring that Malinois who likes to kill cats and let them do their thing? No, that doesn't work. But it was a huge systematic traps and all sorts of short life poisons and things like that to get rid of them. But they were successful. We will continue now with the boats, but also to survey other satellite islands in that area. And in every case, they say, we think there may be a couple of rats there, but they're just right around the port. And then you take the dogs, and they are just around the port, and up the hill, and over the hill, and around the side of the island. And they're far more widely distributed and that makes sense, right? Because dogs are so good at detecting at those low densities when they're just getting started. So I'll tell you about some of the other, these other sort of examples as well. This is Lily again. That's Lespedeza, Chinese bush clover. It's at Neil Smith Refuge in Iowa. Lily's been doing that with Amy for how many years now, Amy? 5 years about. Interestingly, at the early part of this project, Lespedeza was all she looked for. But now, well, let me back up and tell you an interesting thing about dogs and the way they search, which is that they search differently than we do. We have a search image. We're very visual. So if I tell you there are dollars hidden all over this room, right, you're thinking of paper dollar bills. But if I hide silver dollars all over as well as paper dollars, you will be less effective at finding them because it's harder for us to balance two search images, or it's an image that you're not actually looking for. That doesn't seem to be the case for dogs. Dogs can search for multiple targets at the same time. And so in the last couple of years, Lily has started looking for rare and endangered host plants for monarch butterflies while she's looking for bush clover. So one of the things, and I think we'll get into this when we get into the discussion, one of the things that we really encourage projects and people interested in dogs to do is to look for whatever else is of interest there. So when we take dogs all the way around the world to Nigeria or Cameroon to look for cross-river gorilla scats, we should also be looking for golden cat or leopard or many of the other species of interest in those areas. And this is a great example of doing that. I'm going to spend a little more time telling you about Dyer's Woad because it's a cool story and it's one that I think may resonate with all of you guys. Dyer's Woad is that plant. It was bred or brought to our part of the world to make dye, and it's a classic. You guys probably know it, but it makes a lot of seeds, and it gets into places and it can take over. This is not Montana, thankfully. This is Utah, but it'll cover whole hillsides and take over. So when it arrived in Montana, people were justifiably freaked out. And it arrived, fortunately, I think, right next to the University of Montana, the hill, Mount Sentinel, where the M is, if any of you have been to Missoula, and hiked up there. It arrived there. 99 was when they started counting it. 2004 was when they started counting it and making note of its status, whether it's flowering or reproductive versus not. And you can see it was going gangbusters. So they really made an effort to start knocking it back, and they had some success. They were pulling hundreds, thousands of plants, but you can see they got down to an area where they just kind of plateaued, and they were having a real hard time knocking it back any further. And that's when they talked to us and said, can you bring in the secret weapon? And Seamus is that secret weapon. He's one of them. There are a number of dogs. He's our current guy. He's a border collie. He flunked out as a ranch dog, and he's a very intense guy, if you can't tell. in that picture. He trained on potted plants, not reproductive, and as usual, we do a lab study. Can they detect it? They do, and then they head out to the field. And so Seamus and the other dogs have been working on Mount Sentinel. It's a big, grassland. It's steep. When it gets wet, it's like greased bowling balls on that hillside. And detection distances are not huge, so it takes a very intense search effort. So what you can see, there's the M up there. There's campus down at the bottom, and those little lines are the tracks, GPS tracks. It's an intense search. They search the whole area. It's not sampling. They're trying to get every last individual. So the bottom line is it's working, right? They are knocking it back. And the most important part of this figure is the reproductive bits. That's the secret to doing it, because I mentioned what a crappy botanist I am, but even good botanists often recognize and see the plant once it's flowering. And as you guys know, once it's flowering, it's too late. Dyer's woad, 12 to 15,000 seeds, they say. So it's already dropping seeds, and you're behind the curve. But Seamus doesn't need that. So this was, I think, 2014. These are the kinds of plants that Seamus is finding, as the other dogs too are finding on Mount Sentinel in this kind of cover. You'll never see that. You just can't search visually for that kind of thing. It often takes the handler minutes to dig around and find, and the dogs have a secondary command, which is show me, and they'll show you what they're actually smelling, because often you can't see it. And we use that for all sorts of different things, but this is a pretty important context for that. And this is one of even the most impressive ones. And you can see he's off. He doesn't care about Dyer's woad. He cares about his ball. He's playing with his ball while he's being rewarded for finding that. So that's the kind of things that they're finding on the mountain now. So we're down in this tail now, and I'll just kind of explode this end and show you the data for the last couple of years. Our take on these data is that there are so few individuals. Usually when they record an individual as reproductive now, it's just starting to have buds. So the hypothesis and the hope is that we are just working through the seed bank now. We're waiting for everything that's in the seed bank to come out. Hopefully we're not putting anything new into the seed bank. Interestingly, 4 individuals in 2017, they were all in 4 different patches. We know the areas where it could be now. This year, over 20, 25, partly because we had an exceptionally wet year. So snowpack in certain parts of the state was 300% of normal last winter. So hopefully it's because it's really wet. Maybe that water's penetrating more deeply. Who knows? Maybe this is a big event. It's going to get some more of those seeds out of the system. So stay tuned on this one. This is, and this is another interesting institutional problem, is because it's not overrunning the mountain anymore. It's tough to keep the funding coming for this. And even if we have to do it on our dime or eat a loss to do it, we're going to do it until we get to that, a number of years of zeros, so that we can say it's been done. Now, hopefully, won't be reintroduced, but the 3 key things that made this possible was, one, they were just better at finding them than people were. The first years that we did this, it was an experiment, and human searchers would go out, and they would bring the dogs after human searchers had said, that's all we've gotten. The first year that they did that, human searchers missed over 500 plants. Now, there aren't 500 plants out there, but so they're finding these, these inconspicuous ones down in the leaf litter and in the grasses and all that stuff. But the real key to this is the earlier detection. They get it before it's flowering. So that's really cool. There's also a good story in here that was about the biology of the plant. There was a dog named Weibo, who was one of the early dogs on this project, who kept going back to the places where plants had been removed. And the thought was, well, he's just driven. He just wants to get rewarded. There must be some residual scent there or whatever. Well, sure enough, some of those spots where plants had been removed resprouted. So they didn't know that it would resprout from just a root fragment. So now, based on that revelation, now what they do is if it's reproductive, they cut off any flowers, and then they flag it, and then apply a contact herbicide just to that individual plant, and then they come back a week later once it's dead and dig it completely out. So the dogs actually helped us understand that we didn't understand what was going on with the plant. Okay, I mentioned the Carter Cruz, who's Ted Turner's aquatics person, you know, on all of Ted Turner's properties in the West. Carter runs the aquatics program for all of those ranches, over 2 million acres. And they do very cool, interesting work. Part of that was eradicating brook trout from one reach called Cherry Creek that's close to Bozeman. A little bit controversial, but Yellowstone, the idea was take out the brook trout, yet let native Yellowstone cutthroat come back in that area. He did it. It was successful. It was very controversial. You guys know electrofishing. This is how they do it. It's costly. It's a little bit dangerous. It can be. It can be unpleasant when you're doing it that time of year. But you know, most sort of importantly, it's slow. It is really slow. It takes a long time, particularly if you want to find one fish. So Carter said, can dogs do it? And we said, well, they find cadavers underwater, right? There are a few good examples of dogs finding grizzly scats and other things underwater. So we said, well, let's try it. So we have a place in Bozeman. Back in the day, it was a fish hatchery. Now it's called the Fish Technology Center. They breed endangered fish and do all sorts of stuff there. It's U.S. Fish and Wildlife Service facility. So we went there and electrofished, caught a bunch of rainbows and brook trout, swabbed them, took a swab and just wiped it on the fish, and then trained the dogs on those swabs. So the first question was, can the dogs tell a brook trout from other species of trout? And as Tia is telling us there, yes, they can. They were able to discriminate fish to species based on the swabs. So the next step was at the Fish Tech Center there, they've got these experimental streams that they can portion off and do all sorts of stuff like that with. So this is Pepin. He's a Belgian Malinois. And he and 2 other dogs ran up and down and told us which of those chambers had brook trout in them. Some had brook trout, some had rainbows, some had nothing in them. Sure enough, they can do that. So he said, awesome. So we took them out to the field, and just under the bank there, you can see that little silver. That's just a little fish trap. Carter, that's Carter and Carter was very skeptical about this whole process. And between us, he was really puckered because we're taking brook trout out in those little fish traps. He you know, back to the stream that he had eradicated. It was a lower portion, you know, and that hadn't been controlled. But he's like, don't let these fish get away. And so, sure enough, we put the dogs in, and it was an amazing moment because this little stream is running downhill, and there was just a slight uphill breeze that day. And so what was happening was the scent was coming out and kind of piling up, and the breeze was holding it and piling it up. So when the dogs came up the stream, they hit it like a wall, and they just went bam. And Carter, who's standing there, you know, with his clipboard, you know, everybody's trying not to signal or cue the dog or anything. In fact, this was double blind. The handlers didn't know where the fish were or which traps had fish in them or not, everything. And the light bulb went on, and they were able to do it. So they were, and discriminate both brookies and rainbows, and also empty, and didn't alert on the empty traps. So they're not just visually cueing on it or cueing on our scent on there. What was really interesting about this one was each dog did it totally differently. Pepin was in the water. He was biting it. He was slamming around in there. This is Rue. She's a young dog. She was in training. She just kind of gently walked up there. And Tia, the old shepherd, she just walked along the shore and kind of dipped her nose in like that. We don't know how they're actually doing it, you know, or they each have individual styles, but they were all able to do it. So this is a big one that I would like to see us continue because there's lots of applications for it. We need to go the next step. We're working with a fellow named Adam Sepulveda at USGS, who's doing a lot of very cool eDNA work, and we're going to try and figure out what's the biggest stream that we can do this. Could you do this in the Missouri? You know, flow rates, what are the constraints? Obviously, if it's deep and fast flowing, if the scent starts here, it's going to come up way up there. So that may limit how useful it is for us. But but we'll see. He gets some very strange eDNA results. Often he'll get positive, 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 and then nothing for a long stretch, and then another positive way up. So his interest is to use dogs to fill in some of those gaps in the areas in between to say, are we detecting just separate populations or aggregations of individuals? What's going on there? So I'll just mention a couple of little things to end here. One is, you guys know this, but everybody loves dogs. Everybody loves a dog story, right? The press come out of the woodwork whenever there's a dog around. So almost all of our work is in partnerships with folks like you. And we work with nonprofits and agencies and all over the world. And the story that always comes out is, I've been slogging away trying to get people interested in whatever it is, this species or this issue, for years and years. And then the damn dog shows up, and all the press are here, and they want to hear all about it and everything. So we now take that very seriously and say, this is an opportunity, and you should seize that opportunity to do press days and to use it as a way to get your message out. So that's Pepin. He was working on law enforcement ivory there, and there's a PBS documentary just made. Amy had the privilege of going to the Bass Classic Championships in Texas. And that's Salvinia Man with Wicked and Lily there. They reached 50,000 people in just a couple of days doing all that work. And of course, there's Pepin again. He was with Jane Goodall. And of course, the big hall filled with lots of people, and Jane comes over to the dog. She doesn't want to talk to everybody else. She hangs out with the dog. So it's a huge opportunity. And our friends in Alberta, Amy has been part of training 3 dogs to go to Alberta, and they are the model. They have really seized this opportunity far greater than anybody else. Is it going? May not. There it goes. And basically they saw this coming, and these 3 dogs were trained on zebra and quagga mussels early on. Cindy Sawchuk, who was hugging Hilo there, is the lead for Alberta. And basically, these dogs are really good at sniffing mussels, and they're really, really good at engaging the public. So they were getting so much press that they started doing YouTube videos like this, and they have had the Minister of Environment for Canada out, and Hilo was quite rude to her, jamming his nose where it shouldn't have been during the press conference on stage. And she still said, we're going to renew this program. We're going to have a full-blown program. So Cindy, after a lot of this press and interest and all of that was happening, Cindy asked her public relations person, she said, how many people do you think we're reaching? And they have good metrics on all this stuff now. And she told her, and then she said, how much would it cost us to reach all those people in a traditional education, outreach, whatever campaign? And in the first year of the program, it was over $1 million Canadian that they got for free just because these dogs are, they're rescued, they're great stories, and they're cute, funny dogs, and people get out of their cars. The same story with the inspectors are saying, whereas people are really unhappy to see me, now they're getting out and they're saying, oh, who's this? And they meet the dogs and they play with them, and they're engaged. They're happy to have them search.
Why are they wearing boots?
Sorry?
Why are they wearing
for scratching the boats, so that they don't scratch people's boats. Some people, can be very concerned about their boats, which is fair enough. So it's been a great program. And now these dogs are now, for the off season, their big time is when the snowbirds come home, April, May, when everybody comes from the south. And so the rest of the year, they're less busy. So these dogs are now trained on thesium, an invasive weed in Alberta provincial parks. So it's exactly the kind of program. These guys are the model program. We think they're just doing wonderful stuff. So, this is what we want all of them to end up being like.
I was just going to interject a good story about how they can't, people have to pull over for these mandatory inspections, and they do it all the time. It doesn't matter if you're in and out. There's commercial guides that are doing a loop all day. Every time they go through, they have to pull over for inspection. And so the dogs can just, bring a kind of a nice relief to that. And, have a team working in Texas right now, and there's a tournament bass fisherman, really fancy, nice boats, and the Park Service is asking him, have you been inspected before? Yes, and those people have their hands all over my boat. And the inspector said, well, we have the dogs here today. Can the dog inspect your boat? Oh, yeah. But that's when, the little boots come in handy to make sure that this fancy boat stays.
Yeah, exactly. Okay, so this is a bit of a segue into the much more practical discussion, which is if you actually want to do this, what do we need? And these are, the discussions often go, can a dog do this or can a dog do that? And we go, yeah, probably. And then they say, and then it's often, is this the most cost-effective way? And then the logistics of actually doing it are really the challenging ones. So in order to train a dog on a specific species or target, we obviously need to have some of those. And in the scat world, right, we need scats that have been confirmed, that are of known provenance, right? So you have to train them on something that is absolutely certain that it's a grizzly bear, right? And then you also, if you're going to reward them in the field, you need to be able to confirm that that's what it is when you're in the field. And I'll tell a story that came from California. Thankfully, it wasn't our organization who was involved in the work, but they were working on Pacific fisher, rare species. Happens to co-occur with pine marten. And the handlers, who were not very experienced, are walking through the forest, and the dogs start to get bored, and because they're going long periods without an alert, right? And that means without a reward and without getting to play ball. And so the dogs will sort of come across a pine marten scat, and they'll kind of give a little halfway alert, like, hey, I'm just gonna try this and see if you'll go for it, right? An experienced handler says, yeah, nice try, buddy. An inexperienced handler says, wow, we've been walking a long time. He's working really hard. I'm just going to reward him. Well, you just trained that dog on pine marten, right? And $140,000 later, the Forest Service was very angry that their Pacific fisher genetics budget just got wasted trying to get fisher DNA from pine marten scats. So you have to be disciplined. You have to be very disciplined in the field. And that means discrimination in the field. If you can't discriminate it in the field, it's not a deal breaker. It doesn't mean you can't do the project. It just means you have to do it in a different way. And it's a lot more work, and you basically have to trick the dogs and treat them like drug dogs, right? And set up successful hides for them where you know what it is. So it can be done, it's just harder. Amy's working on some new methods and a different paradigm and a different way of training dogs that may help us work around that sort of stuff. But for the time being, these are a lot of the things that you'll need if you're going to be able to do a successful project. The other thing, the things that give me heartburn when I think about this stuff is, what do we train on? What's not here right now? And I went to a conference in Seattle a number of years ago, and there were all these predictive models about invasibility and the life history characteristics and the characteristics of communities that would be invaded and all of those things. And at the end I said, okay, so what do let's say we just brought every one of our dogs and they're here. What are we going to train them on? That's a hard sort of question. And I have nothing to offer for you guys in that context. In the case of zebra and quagga mussels, when you've got this marching advance coming, that's an easy one. What's the next thing that's going to come out of the ballast water of some boat that's coming in from who knows where? It's a hard one. Yeah, that's a hard one. So I'm interested to know about those things from your guys' end. And then, I often, it's amazing. The sort of, what would you call it? The sort of noise to funding ratio is exponentially higher with invasive species. We get people saying, you've got to come work on this, you got to do it, you got to do it. I say, awesome. Is there any money? No. There's $1,000. Or something like that. And it's really, we do send dogs out where we can, when we can, I think, but to do it right, it takes a tremendous amount of vigilance. You got to be there all the time. 1 in 1,000 boats, right? You got to hit a lot of boats to do that. We can't just shoehorn that in with the rest of the day job. So finding the money can be really a challenging thing. The last thing is these links that we talk about, the links between detection and action. A lot of times we'll be asked can a dog detect this? Can you take him out to the field? And the answer is, and then what? If a dog alerts, what are you going to do next, right? And that's a big question. And a lot of times, one of the challenges is people are very interested in having dogs and not necessarily willing to make the full commitment to use those dogs to maximum effect. Alberta's really invested, and they've gone all in on those dogs, and they're doing a great job. Unfortunately, Montana, despite allocating $9.5 million from our legislature, has still not ponied up the $30,000 to train dog teams, to have them on the ground all the time. And I would say that's probably part of the reason we got the positive result. And it will be the reason that we get them eventually, if we don't pivot and commit to them. So it's an ounce of prevention, and it's dirt cheap compared to getting mussels in those waterways, and yet it's still a difficult sell. So we were just out with Zach doing the shoreline surveys, and we're about to have a legislative session in Montana. I said, take that message back. Tell them how expensive it'll be, and tell them how cheap it is to have the dogs, for prevention. And you can repurpose them all over. And particularly if you can do them for multiple species, you can, cobble together a full budget. My contention is that every state agency, everybody ought to have dogs, and they will in the next 10 years once it's a lot more well understood, what they can be used for. We've got dogs that do endangered species, law enforcement, and invasive species. Those aren't different things. It's all playing ball as far as the dog is concerned. So some of them are different. Some dogs like to do detail work. Some dogs like to run big and do other things. Some are spastic and moving all around. Some are very quiet and slow and do their thing. So there are individual search styles that make a dog better or worse, but there's nothing inherently different about searching for invasive species or law enforcement or endangered species or whatever that would prevent a dog. So Amy, one of her personal dogs that just retired was named Wicket. Wicket was the most experienced conservation detection dog ever. She was on 32 different scents when she retired. And those scents are all over the world, and she'll look for any of them, all of them, anytime. So I encourage you guys, as we have this conversation, to think broadly about what the applications are, and particularly how you can cobble these things together and make them sort of cost effective. So Carrie, should we all sit up here, and maybe I can answer one question while Josh and Linda and Amy, we all sit up here and
Sure, yeah, and I just wanted
answer.
to kind of transition to, and you can definitely answer us a couple of questions
Sure.
if you want right now, but we are going to have a panel and a chance to ask a lot of questions.
Yeah.
So why don't we say 2 for now, and then
Perfect. Go ahead.
Okay, cost, for training and maintaining.
It costs us about $10,000 to rescue and base train a dog. And so I didn't mention this. We rescue the dogs that we train. There are lots of good dogs in shelters. We have a nationwide sort of network of ways to find them. We screen about 1,000 dogs for every one that we take. And then about half of those wash out during training. So it's $10,000 roughly to take a new dog and train him to be a detection dog. After that, it's really easy to add new scents. Once they learn the game, they're good at, they figure, they know immediately, oh, right, okay, I'm going to add this to the things I'm looking for.
What about their handler?
They're, cover your ears, Josh. Yeah, don't take this personally, Josh. It's way harder to train the people than it is to train the dogs. And Josh is already a handler, he knows. Yeah, and we do that. We don't sell dogs. We don't just take a dog and train it and sell it, because the day that dog leaves us, it's a great dog, and it starts to go downhill from there. Dogs are never finished. They're either training or they're retraining. And so we do a ton of updating and support and all that. So Amy's probably, was it twice a year now? Works directly with those Alberta teams. Our teams in Zambia, we're on WhatsApp and everything, weekly, and we're in Zambia and Tanzania. Well, Tanzania still, because it's a newer program, 4 times a year, helping train them. So anywhere from, it's about $5,000 per dog per year to support them, to keep them clean and working well and working productively. And you know, there's the technical side of things, the detection and the discrimination, and then there's the behavioral side of things. You know, Seamus just developed this, he would come unglued, you know, cars and people and big dogs, and, you know, and so some dogs who do endangered species work, they're out in the bush, who cares? They, you know, they can be as antisocial as they want to be. But, you know, particularly like with all of you doing inspections and things like that, you need dogs that are going to be good with the public and all of that. So it takes some behavioral upkeep as well. So often that $5,000 per dog happens, you know, in that way. And, you know, for those of you who don't know, we're a 501c3. You know, most of my time is asking other people for money. So we jointly fundraise for those sorts of things too. So particularly when there's a strong conservation angle and things like that, it's very easy for me to go out and get money for jaguar and elephants and rhino and, you know, things like that. You know, the more heady it is, the more sciencey it is. It's a little harder to get individual donors to go for it. But we often work with our partners to fundraise for that sort of that side of things.